The first presentation of this session will be presented by Aparajita Mitra, PG student of AIIMS. The role of fiber optic phototherapy versus in less harmful to the rat germ cells and conventional phototherapy.
Good morning. Thank you for this opportunity to share the results of our study, which was conducted at AIIMS. So this is based on a pilot study which has been published in Pediatric Research 2014, which showed that conventional phototherapy causes histological changes in neonatal rat testes. So the aim of this current study was to compare neonatal conventional versus fiber optic phototherapy on testicular germ cells in rats. Now we divided the mother rats and the pups into 3 groups, of which group 2 received 48 hours of conventional phototherapy and group 3 received fiber optic phototherapy for 48 hours. After weaning, 6 male rats per group were sacrificed on day 70, day 100 and day 130 of life. Now the testicular tissue was subjected to multiple studies, including histology for the parameters such as seminiferous tubular diameter and the Johnsen testicular maturation score. DNA flow cytometry was also done to study the haploid cell population in order to find out the maturation of the testicular tissue. Now these are the photomicrographs of the 3 groups at 70, 100 and 130 days. The seminiferous tubular diameter was found to be significantly different amongst the 3 groups at 70 and 100 days of life. Now this is a morphometric parameter. And the Johnsen testicular maturation score was also found to be significantly different amongst the 3 groups at 70 and 100 days of life. DNA flow cytometry, you can see the 3 peaks which denote the haploid, the diploid and the tetraploid cell populations. This was found to be significantly different amongst the 3 groups only at 70 days of life. So to the best of our knowledge, this is the first study of its kind to study the effects of conventional versus fiber optic phototherapy at the appropriate ages of maturity in the neonatal rat testis. And it is also the first study to compare the 2. Now, the take-home message from our study was that only conventional neonatal phototherapy produces a short-term adverse effect on testicular germ cell maturation. The natural question in our minds would be, can this be extrapolated to humans? Does exposure to phototherapy in the neonatal period cause a delay in puberty after conventional phototherapy? Now the only answer to this quandary would be to conduct further randomized control trials to compare these 2 types of phototherapy. Thank you.
Thank you, Dr. Aparajita. It's a good research pilot study and it may be a future eye-opener for the manufacturer of phototherapy units.